Hello everyone, in this video we are going to learn about biosimilars. To understand biosimilars, first you need to understand what are biologic drugs. When you think of drugs, you often think of traditional drugs like aspirin, a kind of pill that you can pick up from your local medical shop or pharmacy. But biologic drugs are totally different. They are proteins which are made using living organisms, whereas these traditional drugs are made up of chemicals referred to as small molecules. In addition to that, biological drugs are much larger in size compared to small molecule drugs like aspirin. These biologic drugs or products are produced through biotechnology in a living system such as microorganism, plant cell or animal cells, and these are regulated by the Food and Drug Administration. These drugs are used to diagnose, prevent, treat and cure diseases and medical conditions. Biological products are a diverse category of products under generally large complex molecules. There are many types of biological products approved for use in the United States, including therapeutic proteins such as filgrastim, monoclonal antibodies such as adalimumab, and vaccines which are used to treat influenza and tetanus. Familiar biologic drugs include widely prescribed therapies like etanercept, infliximab, and adalimumab. Okay, now let's move on to the topic. What are biosimilar drugs? Whether they are new drugs? Absolutely no. They are the copies of biologic drugs and have been used to treat many diseases. Generally, each biosimilar is made using the same amino acid starting material and the same step-by-step processes like reference drug. Reference drugs are nothing but the single biologic drug which we are referring to manufacture the biosimilars. At last, the proposed biosimilar product is compared and evaluated with reference product to ensure that the biosimilar product is highly similar and has no clinically meaningful differences. The minor differences between the reference product and the proposed biosimilar product is the modification that we are doing in clinically inactive components. For example, these could include minor differences in the stabilizer or buffer compared to what we have used in the reference product. Biosimilar drugs are basically generic drugs? No. The below mentioned points are some of the differences between biosimilar drugs and generic drugs. First of all, these biosimilar drugs are large molecules, but generic drugs are small molecules. Biosimilar drugs contain complex structure and have large molecular weight which ranges up to 300 Dalton. But these generic drugs are composed of simple structure and have small molecular weight which ranges up to 270,000 Dalton. Biosimilar drugs are isolated from living organism, whereas these genetic drugs are produced using chemical synthesis. And also, this is the most important point, these biosimilar drugs need special approval to be interchangeable product, but these generic drugs are automatically interchangeable.
These are the stages of drug development process. First is comprehensive molecular analysis. It's to identify the reference biologic using various tools like mass spectroscopy. Next stage is biological quality assessment, both in vitro and in vivo assay to confirm the structural and functional assessment. Then in next stage is in vitro pharmacology and preclinical stages is to confirm the tolerability and dosing. Next is pharmacokinetics and pharmacodynamics. This is phase 1. Pharmacokinetics is nothing but how drug behaves to body. Pharmacodynamics is nothing but how body reacts to drug. Next is clinical trials, that is phase 3 for safety and efficacy. Next is final stage is post approval commitments, that is phase 4 studies. Manufacturer must prove that any difference that may occur in no impact on safety and how the drug works. And next is time taken for development of biosimilar drug. It taken 2 years to product development and a comparative analysis, and 4 years for scale up and validation, and 7 years for clinical trials, 9 years for FDA review and approval. Next is we take a monoclonal antibody, that is biological product with different post-translational modification like bisulfate shuffling, truncation and fragmentation, etc. This is the molecular analysis with reference product aspirin. And this is the analysis of relative molecular mass of biosimilar drug like monoclonal antibody. Next is therapeutic indication of biosimilar drug evidence like physiochemical characterization, biological activity, safety, pharmacokinetic and pharmacodynamic profile, route of administration, mono and combination therapy. Biosimilar receives all indication of the reference based on the evidence from comparative studies. Next is the analysis of biosimilar drug approval and approach at every year. Next is precise step of biosimilar drug development, that is characterize reference product, target director iterative development, establishing similarity, additional similarity for information.